In the study of evolutionary developmental biology, it's really important to be able to know when in evolutionary history a trait evolved to answer questions like, did traits evolve multiple times in parallel in different lineages, and so forth. So for the study of Evo Devo, it's really important to be able to understand how to view and interpret a phylogenetic tree. So here's a basic phylogeny. And first, let's talk about how to interpret the structure, the drawing of the relationships between individuals. And in this case, it's common that what this family tree essentially represents, at the tips of the branches are either populations or species. So in this case, we'll draw a species tree. So we might have species A, species B, species C, D, E, and F. So what this indicates is that at this point in time, these are currently existing or extant species. So extant means currently existing. So here we've represented 6 different species. And in a phylogenetic tree, there's always a root, or almost always a root. Some of the phylogenetic trees, which are what these branching structures are called, don't have roots. But rooted trees have directionality to them in that the tips of the branches, again, representing extant or currently existing species, represent the current point in time. And that means that the root of the tree represents organisms that lived in the past. In other words, all 6 of these species have a common ancestor. And that would be an ancestor that is not currently living. It's an organism that is now dead. But a common ancestor gave rise to groups of organisms, lineages, different branches of this tree that eventually, through mutation and evolution, gave rise to 6 different species in this tree. So we've talked about the root of the tree and the tips of the branches. And then there are also nodes. So every time a branch splits into 2 branches, that's referred to as a node. And that point in time essentially represents, well, in the case of a species tree, which again this is, a node represents the place where speciation occurred. Where one lineage became 2 separate lineages. And it's been pointed out, and it's important to note, that speciation does not happen at one instance in time. That is, speciation takes a long time. So although time is shown as a linear variable, which it is on the x-axis, speciation doesn't happen instantaneously. It takes a stretch of time. It's just easiest to diagram these branches as having a clean single point where they split, but that's not actually biologically true. So we've got the root, we've got nodes, we've got branches. And then the next thing, the most important aspect of interpreting a phylogeny in its most basic form is understanding the shape or the structure of the branches and how this tells us something about the relationships between different species. So for example, here we have a common ancestor. So a common ancestor is defined as the organism that gave rise to all of the species that are being discussed. So in this case, in black, the common ancestor of all 6 of these species, because all 6 of these species have lineages that trace back in time to that common ancestor. But there are multiple common ancestors in this tree. For example, there's also a common ancestor of species B and C that lived more recently in the past. So each node represents a common ancestor to branches or branch tips, species in this case. One of the things that is really important for interpreting phylogenies is understanding the relationships between different species, different branch tips. One pattern is sister species or sister taxa, because taxa can refer to more than species. It could refer to genuses or genera. If this was a genus tree, it could refer to populations. Individual taxa could be individual populations. So it's a more generic word than species for the purposes of phylogenetics. So those are sister taxa. They are the most closely related two species to each other, and that's because they share a common ancestor most recently in the past. This defines those two taxa, those two species, as a clade, which is defined as the entire set of currently existing taxa that share a common ancestor and not leaving out any other species. So for example, to make that point a little bit more clear, B, C, and D is also a clade because that group of three species also shares a common ancestor. But we would not call C and D a clade because although they share a common ancestor, back here at the node in blue, this is not a clade because we're omitting one of the descendants of that common ancestor, which is B. So the clade is all of the descendants of a common ancestor. So in this case, B and C have a common ancestor here. And so all of the descendants circled there in yellow make a clade. C and D is not a clade because this clade from this common ancestor would have to include all three of those species to be called a clade. And these relationships between B, C, and D bring up a really important concept that is how to measure, how to read a tree to determine which species are more closely related to each other. So in this case, sister taxa, any group that has a common ancestor most recently in the past, also here in the pair of species E and F, are again referred to as sister taxa. They're the most closely related species on the tree. What, though, is the next most closely related species to B? C is the most closely related because they shared an ancestor recently in the past. The next most closely related species, you go back in time to the next common ancestor and look at its descendants. In this case, that would be D. B has its next most closely related species, aside from C, after C, is D. And then the next most closely related species from B, after C and after D, we go back one more step in time and find the common, or the descendants of that common ancestor, which would be A. So the closest relative of B is C. The next most closely related species is D. Then the next most closely related species is A. But now, what about, again, compared to B? Is E or F the next most closely related species on the tree? This is a little bit tricky, but the overall goal here is essentially the length of the branches, the actual distance that the cursor travels, or your finger travels, or your eye travels to get from one node, or from one branch tip, one species to another, is the relationship of the distance. In other words, B, even though E is drawn on the tree, and this is somewhat misleading, but because E is drawn physically closer to B on this tree, a lot of people tend to think that E is more closely related to B. That's not exactly how this interpretation works, because if you travel from B back in time to the common ancestor of E and F and B, notice that once you get to this node, the distance from B to E is the same as the distance from B to F. So these two sister species are equally distantly related to species B. And I should make one more point here before moving on, which is that phylogenetic trees don't necessarily include all of the species that are known. It just includes species that are being currently studied. So in other words, there are likely many other species that have different relationships in this tree. They're just not shown. So whenever we're talking about the interpretation of a tree, There are a few other examples. Whenever we're talking about the interpretation of a tree, we're only limited to the species that are currently shown on the tree. But it is important to realize both, one, that there are probably other species that just aren't shown or diagrammed. The other thing to keep in mind is that this is not an exhaustive list of all of the species that ever existed. Again, these are just diagrams of species normally, just diagrams of species that are currently existing, the extant species, species that currently exist and we can measure, we can view, we can collect data from. But what's not shown on phylogenetic trees then are extinction events, lineages that existed, but they underwent an extinction event some point in the past, so they are not currently extant species. That's just one thing to keep in mind about phylogenetic trees. They only contain lineages that exist in the present. So here is an example of a vertebrate phylogeny. It has members of different lineages of vertebrates. Certainly, again, not all of the species that are vertebrates that are currently existing, just a selection of them. We're not looking at lineages that went extinct. We're not looking at all extant species. And in fact, some of these are not species, but groups of species like sharks, ray-finned or bony fishes, and so forth. Some of these do represent individual species like the coelacanth or the platypus, the opossum, and so forth. One other really important thing to know about the study of phylogenetics is that this diagram of the relationships between the species is a hypothesis. That is, we don't have a time machine. We can't travel back in time and watch lineages diverge. We can't go back and see these speciation events happen. So this graphic, the phylogenetic tree, is a prediction of what happened in the past. It's a hypothesis that can be tested. It's based on data, but it is not itself fact. Another important point to bring up here, the purpose of this slide, is to discuss how these relationships are inferred. Again, we can't go back in time and actually observe these lineages diverging. We have to make informed guesses about what happened in the past based on information we can collect today. And sometimes that information is data from the past, like fossil record information and ancient DNA from samples that are in extinct lineages or from extinct individuals, dead individuals, anyway. There are a few different types of data that scientists use to infer phylogenies. An easy one are, well, any of these are traits. So it has to be something about an organism that we can observe. Often, this is morphology. How similar organisms, different lineages, different populations, different species look, how they act. And another trait that is very frequently used is DNA sequence or genetic information from each of these species. Both of these and many other types of traits, but I'm going to focus on those two for today, can be used to infer relationships between organisms. So for example, in this tree, we could ask which of these different species have hair? And so hopefully you know where I'm going with this, which is that mammals are defined as a group of organisms that all have hair. And in this case, we also have monotremes, the platypus, the weird sort of ancient mammal, which we can see here on this tree. This is the group of mammals, right? Crocodiles, lizards, birds, frogs, and fish are not mammals. Platypus, the monotreme, and opossum as an example of a marsupial have hair. And then we've got elephants, manatees, primates, including humans, rodents, cow, whales, hippos, which are all closely related to each other, bats, horses, rhinoceros, and cats and dogs. Check out the branch of platypus. If we travel back in time to its common ancestor, the platypus, the monotreme, an egg-laying mammal that's also venomous, has a common ancestor with the opossum, but the platypus is the most distant relative. Actually, it's the closest relative among all the rest of these species. It's the closest relative that's not in a placental mammal to all of the placental mammals, elephant through the cats and dogs. So again, these are just examples of the sorts of traits that traditionally phylogeneticists have used to group organisms into who is more similar or more related to who. Right? So we've got the placental mammals. They all share a common ancestor there. And then we've got organisms that have hair, which I'll switch to a different color, red. So that means that hair in the mammalian lineage evolved in that ancestor. And we're going to get to how exactly I'm making those sorts of interpretations in just a minute. But the point again here is to reinforce the idea that the traits that are used to infer relationships between organisms can be anything. In this case, I've talked about two morphological traits or two developmental traits, the development of hair and the fact that the placenta is involved in the development of an embryo. So again, these are traits that phylogeneticists have traditionally used to group organisms into clades. So we have a clade here. All of the descendants of this common ancestor have placental development and birth from placenta or embryos that develop in placentas. And so that is a clade. That is an exclusive group of species that share one common ancestor, and no species in here don't have placental development. Likewise, the group of organisms that contain or develop hair is also a clade. It's all of the species that descend from this common ancestor. Now, there is a problem with using traits to infer relationships, because some traits aren't the same because all of the species derive that trait from a single ancestor. So because all of these species evolved from an ancestor that we infer had placental development, they all share that trait. But there are other traits, which we'll look at in a minute, that don't fit that pattern. They're not informative for phylogenetics because they evolved multiple times, excuse me. So we're going to take a look at those examples in just a minute. For example, let's take a look at the evolution of flight. Which of these groups of organisms that are shown in this phylogenetic tree can fly? So we're going to ignore flying fishes. That's not really powered flight. We're going to ignore flying squirrels, which is really gliding. So we have birds. They fly. We also have bats in this group that have evolved powered flight. So the principle of parsimony suggests that the fewest number of evolutionary changes is the most likely one. In other words, it's likely that the common ancestor of all of these species did not fly. And then as time proceeded from that common ancestor that didn't fly, sharks eventually evolved, bony fishes evolved, the coelacanth, amphibians, frogs, lizards. That suggests that in this common ancestor between crocodiles and birds probably didn't fly. The evolution of flight probably occurred somewhere since the common ancestor of crocodiles and birds, somewhere on that short branch, after the most recent common ancestor of crocodiles and birds, and after those lineages began to evolve, flight evolved. Before the two lineages diverged, flight evolved. Likewise, the common ancestor of mammals gave rise to lots of species that don't fly, and only a small number of species, bats, that do fly. So that suggests that flight evolved somewhere on the branch from which the common ancestor of cats, dogs, horses, rhinoceroses, and bats diverged. So that's the principle of parsimony. Fewer changes is more likely. Actually, I guess that's fewer changes are more likely. Because of course, evolution depends on random mutations, and random mutations happen all the time, but it's rare to have a set of mutations or even a single mutation that enables a new trait. So it's more likely that this trait would evolve independently, rarely, rather than commonly. So let's take a look at what the other possibility would have been under parsimony. The other possibility is that the common ancestor to all of these species was a flying species. So in that case, that would mean that the reason that sharks don't fly is because they lost that ability somewhere since they shared a common ancestor with that root of the tree. Likewise, ray-finned fishes would have had to have lost that trait of flying independently, and coelacanths, and amphibians. And then on this branch leading to birds, lizards would have had to have lost flight, crocodiles would have had to have lost flight, platypi, opossums, elephants and manatees, primates and rodents, cows, whales, and hippos, that lineage, not each of them independently. And then the lineage leading to horses, cats, dogs, rhinoceroses, and so forth. So which is more likely? It's more likely that flight independently evolved twice, specifically in the lineage of birds and specifically in the lineage of bats, and that the ancestor did not fly, than the other proposal or hypothesis, which is that the ancestor of all of these species had the ability to fly. And then each of these lineages that don't fly independently lost that ability. That is the concept of parsimony. And this really exemplifies why it's important to have an accurate phylogeny to be able to infer when different traits evolved. Because the branching patterns and the relationships between species make all the difference in interpreting whether or not flight evolved once or twice. Case in point, what if we had only been using the trait, because this, I didn't mention, this phylogeny that you're looking at now is based on DNA sequences. But what if this tree had been drawn based on another single trait, like the ability to fly? This tree would look very different. It would have all of the non-flying species on one branch. And on a second branch of the tree, there would be birds and bats. Birds and bats are not the most closely related species to each other. They are not sister species. One is a mammal, the other is not. They're both vertebrates, they both fly, but the trait of flight isn't a good trait to build a phylogenetic tree with because it evolved independently. And if we didn't know that, which we didn't years ago, some scientists in the past didn't know that. But now we do know that birds and bats are very distantly related to each other, as shown on this tree. But if we had drawn the tree the way we would, based on just a single trait, whether an organism can fly or not, this is the tree we would draw. And then we would make an erroneous assumption that flight evolved once, and that the reason that birds and bats fly is because they descend from that common ancestor that already had that trait. So that introduces again the concept of parsimony, how traits can be interpreted and when they arose based on the traits of current species, and critically, the relationships we think they have with each other in the past. And that's why having accurate phylogenetic trees is critical to the study of evolution and including evolutionary development. And so, traits don't always agree. Some traits tell scientists particular patterns of relationships between species, but they don't always give exactly the same evidence of a particular pattern. Some traits will tell you one shape or structure of the tree. We call it topology of the tree. And other traits will give you different topologies. And again, this reiterates the idea that this tree is just a hypothesis. It's not fact. So depending on the trait you choose to draw the tree with or to infer relationships between species with, you'll get different shapes of the trees and different relationships, especially between who's exactly a sister species of who. For example, are crocodiles really sister species to birds, or maybe they're actually closely related to lizards, more closely so than birds. That's not the case. But when you see these sorts of close branching patterns, that is a possibility. That if we had chosen a different trait to study other than genetics, which again was how this tree was built, we would probably see a difference here. For example, birds fly, lizards and crocodiles don't. So maybe that means in itself that lizards and crocodiles should be sister species and not crocodiles and birds being sister species. The point of this slide is to introduce some important vocabulary terms related to the analysis and interpretation of phylogenetic trees. So there's a lot more information on this slide. We're going to go through it one step by one. It looks complicated, and it is, but we'll work through it slowly. We've already seen, for example, the concept of a homoplasy. A homoplasy is one of those traits that's a false trait, a false indicator of relationships between species. And I've listed 3 of them here. We've already talked about one, the presence of wings. So again, the presence of wings, birds have wings, bats have wings. But that trait doesn't mean they're close relatives. It would, but that's erroneous. So a homoplasy, a trait that's a homoplasy, is a trait that's misleading, that makes us think that the species would be more closely related than they actually are. So those are the traits we want to avoid using to build phylogenetic trees. Homoplasies are bad. In this case, there's a group of organisms, pachyderms, is the term for these animals, elephants, hippopotamuses, and rhinoceros, that were traditionally termed pachyderms a long time ago. Pachyderm, thick skin. That's a trait, but it's a homoplasy because elephants and hippopotamuses and rhinoceroses are not close relatives to each other. So although we can refer to those animals as pachyderms, that does not mean they're close relatives of each other. It's a trait they share, but it's not a trait they shared because they all derived that trait or retained that trait because the most recent common ancestor of those 3 species was itself a pachyderm. In fact, because of the branching patterns, we can see that it's likely that that trait, if it is a trait really, evolved 3 independent times. In those 3 lineages, it wasn't a trait shared by the common ancestor. The same with wings. Another one that we haven't looked at yet is exclusively aquatic lifestyle. So manatees are mammals that live in the water, whales are mammals that live in the water, and then we've got sharks, so cartilaginous fishes, bony fishes, and an ancestor to terrestrial vertebrates, the coelacanth, which was also a marine animal. So that would be another homoplasy. We might erroneously think that anything that lives in the water is most closely related, but in fact, that's not true. We've got simplesiomorphy. That's another term related to traits. So we're going to start with the terms that define or help us describe the traits that we use to build a tree. Homoplasy is one. Those are the bad ones. Simplesiomorphies are also sort of inaccurate. These are traits that are shared by some but not all of the descendants of a lineage. So for example, the most common ancestor of all of these species led to sharks, fish, amphibians, lizards, up to platypi. All of those are egg-laying or egg-developing organisms where individual offspring hatch out of the eggs. But that is not a trait that's shared by the placental mammals. So that was a trait that was lost during the evolution of placental development. Right, so platypi have eggs, but all of the placental mammals don't. So this is a trait that's shared by a lot of species, and it is shared by those species because the common ancestor probably did produce eggs. So egg laying was in the ancestor most likely. And then the placental mammals evolved the placenta around there. That trait doesn't define a clade. Remember, a clade is all of the descendants of a common ancestor. Here's the common ancestor to all of these species, sharks through platypi. They all have a common ancestor here, but that excludes the placental mammals. So this is not a monophyletic group. which means a group defined as a clade. And that's a taxon label that we have over here. So a clade is also called a monophyletic group, monophyla, one branch. It means that that is a group of individuals that all share a common ancestor based on that trait. So placental mammals is a monophyletic group. There's a single common ancestor to all of the species in that branch. Monophyletic group, also called a clade. We introduced the term clade first, but formally that's called a monophyletic group. And those are defined, again, monophyly is defined by synapomorphies. Those are traits like placenta that are shared among all of the species in a branch because they all inherited that trait from the common ancestor that had that trait. So placenta evolved here. It's retained in all of the species that descended from that ancestor. And so that is one of the traits that we want. We don't want homoplasies. Those are typically events of independent evolution of a trait that might make us think that species are more related to each other than they are, like wings. Synapomorphies are the traits that are shared among species because those species all contained or derived that trait from a common ancestor. Those are the traits, synapomorphies, that we want to build phylogenetic trees with. Not simplesiomorphies. Those are ones where some members of a group, but not all of the members of the group have it. So this whole tree is a clade, but the whole clade does not have the trait of egg laying. Some of the species do, and that's called a simplesiomorphy. So one last point, one more piece of vocabulary that we haven't talked about yet, and that's paraphyly. We've talked about, actually two more. We've talked about monophyly. There's polyphyly. That's where individuals like the pachyderms seem to have the same trait, but they're not close relatives of each other. That is, they all derive from a single ancestor, but not all of the individuals in that otherwise clade have that same trait. So that's what's called polyphyly. These taxa pachyderms are polyphyletic. They're in multiple places on the tree. They don't all exclusively share one common ancestor with each other. And so it's those traits, like thick skin, those are homoplasies, right? Traits that define a clade are synapomorphies. And the last one then is paraphyletic groups. So we've got monophyletic, we've got polyphyletic. Those are caused by homoplasies. And then we've got lizards. So lizards is a group of organisms. So I've grouped lizards and crocodiles together, reptiles in particular, but let's just stick with lizards. They're a polyphyletic group. And I've written over this already, but what it says here is contains common ancestor, but excludes descendant clades. In other words, the common ancestor of lizards, we can refer to a group of organisms as lizards, but lizards is a paraphyletic group. It's not a monophyletic group. And that's because the common ancestor of this entire branch that includes lizards also caused the evolution of placental mammals, for example. We don't call them lizards. So lizards is paraphyletic. They have a common ancestor, but this group excludes other descendants of that common ancestor. That's why this is not a monophyletic group. Lizards is not a monophyletic group. It's a paraphyletic group, as drawn here. And those traits, like egg laying, the traits that lead to paraphyly, are ones that are called simplesiomorphies. Those are the traits that cause the appearance of paraphyly. Again, we've already talked about this idea that egg-laying organisms derived that trait from a common ancestor, but that trait was lost somewhere in the evolution of part of this tree. And so that means that egg-laying is also a paraphyletic group. If there was a name for all of these species, platypi through sharks, if there was a common word we used to describe that whole group of organisms as a group, that would be a paraphyletic group that shares this trait. And all paraphyly says that they share this trait, but there are other organisms that are descendant of that ancestors descendants of that ancestor that don't still share that trait. Now, we just heard that the goal of building an accurate phylogenetic tree is to find traits that are homologous. That is, traits that are synapomorphies. Those are traits that individuals in lineages, different lineages, share because they inherited those traits from their common ancestor. So now I've taken the same tree and I've added a little bit more information. So again, let's walk through this slowly. It's the same phylogenetic tree, but I've done two different things to this now. I've added numbers to each of the nodes. And we'll use those just as a form of reference. And those numbers, by the way, do not have to do with the numbering of positions in this multiple sequence alignment from these different species. So unfortunately, I had to use numbers for both, but please don't confuse them. So these are just numbering the nodes. We can talk about node 1, node 2, node 3, node 7, node 16, and so forth. The other thing I've done, and the idea in this whole part of the discussion, is that we can use DNA sequences as traits instead of things like production of hair, egg laying, thick skin, flight, and so forth. And so what I've shown, these are fake sequences, by the way. Don't use these sequences. These are not from shark, tuna, coelacanth. I've made up these sequences to represent how these sequences, or how DNA sequences from species can be used and are used to build or to infer phylogenetic trees and the relationships between species. So after each of these nodes, I'm noting a distinction between one spot in this DNA sequence alignment that differs between the two lineages that derive from that node. So we'll take a look at that in just a minute in terms of how this actually works and how to read this style of a tree. So to ensure that we have homologous sequences, there are a few things to note about the DNA sequences we use to build phylogenetic trees. We have to know their homologous sequences from these different species, and that's often difficult. So for example, if these were DNA sequences from genes, from coding regions of chromosomes that produce proteins, for example, we would need to know that each of these sequences came from exactly the same gene, the homologous gene, in these different species. So this would need to be a gene potentially, I don't know, like RNA polymerase, for example, a gene that we know that each of these species has, and that we can unambiguously identify as being all genes that are present in these species, and we're looking at the descendants gene sequences from the common ancestor, and that's what I mean by homologous. These have to be gene sequences that are all related to each other because these were in the ancestor all the same piece of DNA, all the same DNA sequence, the same gene. Ways that this gets complicated are, for example, paralogs. If, for example, in bony fish, there have been 2 rounds of genome duplications in the lineage of bony fishes or ray-finned fishes. So this didn't happen in sharks, and it didn't happen in other vertebrates, just the ray-finned fishes. They had 2 genome duplications, which means in most bony fishes, there are 4 copies of every gene that's present in these other lineages. So if we wanted to compare a DNA sequence between mice and tuna, we'd have to know which of the 4 paralogs of that gene to study in tuna, or more likely, we would just not use that gene. But that also makes phylogenetics difficult with fish because, well, we do want to include them in phylogenetic trees sometimes, but how do you choose which DNA sequence to use that you're sure is homologous to the same species in a mouse, for example? So after we select the sequences to use from different species, then we align them relative to each other, slide the sequences back and forth relative to each other to make sure that individual nucleotides in those sequences are homologous, that we're convinced that we know that, for example, at position 11, which is the one that's underlined there in the 11, that the ancestor, we don't know what the nucleotide sequence was, but that these nucleotides are all the exact same spot on the exact same gene and the same part of the same chromosome that descended from the ancestor into all of these species. And that's a major assumption of phylogenetics. And it's not something we can prove because, again, we can't jump back in time with our magical time machine and check. So in terms of building a tree based on a DNA sequence alignment, normally we have computers do this. We don't do this by hand anymore. People used to, maybe people still do, but generally use programs like Clustal to create these alignments and then to interpret them into a phylogenetic tree. And the way that that happens is most commonly neighbor joining. There are different algorithms to detect, for example, to compare two different sequences and to ask how that tells us something about the relationships. But the basic principle is the more similar the sequences are, the more likely those species shared a more recent common ancestor. So let's pick on horse and dog first. We see that horses and canids and felines, so cats and dogs, are in this tree sister taxa. They're not sister species because these are groups of multiple species, but they're sister taxa. So horses and cats, or horses and dogs, should have very similar sequences. That's why the tree is drawn this way. So let's take a look. They are identical, so I'm just going to put a dot every position where the two sequences from those two species are identical. So we see the sequences are very similar to each other except for here, but all the rest of these nucleotides are identical. So 1 out of 18 nucleotides is different. So they're very closely related. And that AT difference, the sequences are the same. If the sequences were identical to each other, no differences, those two organisms would be at the same branch tip because there would be no evidence that they were different. So the presence of a difference is essential. It has to be present for there to be a branching pattern. That's how DNA sequence alignments produce phylogenies. Separation of branch tips means that there is a genetic difference. And in this case, that's what these nucleotides on the branches leading away from node 16 indicate, that in the horse, there was an A, and that in the lineage of cats and dogs in this case, dogs in this example, there was a T. So what these nucleotides, the branches, represent are the diagnostic differences between the two lineages that derive from that node. In other words, if we compare elephants and manatees, they're sister taxa. So we should be able to find a nucleotide where there's an A in the elephant and a G in the manatee. And although there are other differences between the elephant and manatee sequences, we can use that one nucleotide difference, the first one that I circled rather, to represent a diagnostic difference between the two lineages. But again, those two sequences, elephants and manatees, are very similar to each other. They're not as similar to each other as in this example, cats and dogs, but they're pretty similar. So the closer the two pairwise sequence alignments are, the pair of sequences, the closer together, the more closely related on this tree those species will be. If we go look at the root of the tree or the base of the tree, what's happening at node 1? There's a GC difference. Sharks at one spot in this alignment have a G, and the rest of the species, all of these other species, are going to have a C. So let's see if we can spot that one. So that's this nucleotide, where sharks have a G, and every other species on this tree has a C. And that is remarkable evidence that we're looking at homology, that we're looking at a homologous nucleotide, that no matter what species we look at, from tuna to dogs, and humans too, we're in there too, although we're represented in this by rodents and primates, so mammals essentially. We are primates. The rest of the primates, like chimpanzees, are non-human primates. So all of those very different species share a nucleotide in common. And that difference, G versus C, is how we know that sharks in this case, branched earliest, that they share the oldest common ancestor. So in other words, the shark sequence, the whole sequence, looks very little like any of the other sequences. And that's what defines it as basically the outgroup, the most distantly related species to all of the other species, the one we have to go farthest back in the tree to find a common ancestor. Now, if we take a look at this tree really closely, we're going to find that there are two positions where every species shares the same trait or the same nucleotide. And that's at position 4 and position 15, where every single animal has an A, position 4, and a T at position 15. And that's why that AT is present on the root or the base of the tree. And that is good evidence, again, that we're looking at homologous sequences, because every single animal on this tree shares those two nucleotides in common. That tells us something about the fact that we're actually looking at the same parts of the same chromosomes of all of these species. They do actually still have some shared nucleotides in common, even over millions of years of evolution. And those are called uninformative, even though they're actually useful to see. Those are uninformative nucleotides because they don't tell us differences between the species. So even though I've included them in this alignment, most of the time, phylogeneticists would remove any column where every species they're looking at shares a nucleotide, because common nucleotides don't give us information about the differences between the different species. It only tells us that they're related, but that's not what we're trying to do when we look at phylogenetic trees. So I hope that gives you a good sense of how to interpret phylogenetic trees, how to look at where and when, where on the tree and at what point in time traits evolved or were lost, using the concept of excuse me, parsimony, and also to understand how the different traits that could be used to build trees are better than others. So some synapomorphies are much better than others, like homoplasies, and specifically how DNA sequences are used to help infer phylogenetic trees.